Hello, my name is Veglia, and I'm going to discuss the membrane receptors, G proteins, the cyclic AMP, and the signal transduction. What I have illustrated here is the phospholipid bilayer, which is going to be the center of discussion. The upper part of the diagram is going to be the extracellular portion, and the bottom is going to be the intracellular portion. A signal transduction occurs when an extracellular molecule activates a receptor on the surface cell. Here is our receptor. It is a communicator of the cell. It causes the body to react in a certain way. Metabotropic is a class of receptors that includes neurotransmitters. It is going to take any of our neurotransmitters or hormones that come its way. Right here is what I call a ligand. It is going to be your neurotransmitters and our hormones. So the ligand moves over and attaches to the receptor, and it's going to communicate with the receptor. It will tell the cell what it's going to do. It will then produce a cellular response. Now what I'm drawing here is going to be our G protein. It is called heterotrimetric G protein, which will essentially have three parts: the alpha subunit, the beta subunit, and the gamma subunit. These are very important parts in communicating to the cell on what's going to happen next. Now with the alpha subunit basically is going to be telling the cell what it's going to be doing. Then we have the GDP and the GTP. These are both very important because it is essentially say what the alpha subunit is going to do. Now if we have the GDP attached to the alpha subunit, it is going to be inactive and nothing is going to happen. So signal transduction does not continue. So instead, if we have GTP connected to the alpha subunit, then we are going to continue the process. What the GDP is going to tell the G protein is to communicate with what is called the adenyl cyclase. Adenyl cyclase is going to communicate with the G protein. The G protein is what we call a first messenger, and so the first messenger is going to communicate with the adenyl cyclase in order to create cyclic AMP. Real quick, I want to mention that sometimes instead adenyl cyclase will be working with photodiesterate, which is the absolute opposite of adenyl cyclase. So instead of building cyclic AMP, photodiesterate will tear down cyclic AMP. So what I'm doing right here is the ATP. Adenyl cyclates will take ATP and then it's going to create cyclic AMP. So through this process, it is going to create lots of cyclic AMP. It is basically enhancing the response, so there will be an increase in cyclic AMP trial. Cyclic AMP will communicate with a protein kinase, which I am going to draw here. Protein kinase causes an action in the cell. So in order for protein kinase to operate, we are going to talk about protein kinase A, since it's very specific to the cyclic AMP. Protein kinase A is going to cause a reaction in the cell. Cyclic AMP is going to bind to the protein kinase A. There will be a lot of cyclic AMP and there will be a lot of ATP turned into cyclic AMP. The cyclic AMP binds to the protein kinase A and it will activate the phosphorylation process, which is represented here. Phosphorylation is the transferring of the phosphoryl group until an action is produced. So I am going to take the glycogen, assuming that it's a process going to the liver, and it's going to go through the process of phosphorylation, and the cellular response is basically the whole process of signal transduction. The protein conformation will then create glucose. This is the process of cellular response and signal transduction, and with membrane receptors. Thank you.